Good morning, students. Today our topic is about ultrastructure of prokaryotic cell. So it's a basic a microbiology topic or a individual biology topic. We can consider it in any way. So in my experience during the classes, large number of students, it may be a inter students or it may be degree students or it may be PG student. They cannot differentiate the what is prokaryotic cell and eukaryotic cell. So there is a basic differences which I am going to clarify. By that, they in their lifetime they can clear out this problem. That is, what is a prokaryotic one and what is a eukaryotic one? What are the basic differences that are existing in both the cells? By coming to that, prokaryotes. Prokaryotes are nothing but prokaryotic cells. Prokaryotic structures. That is, if a cell having a true nuclear membrane, then it is divided into two compartments. One is cytoplasm and another one nucleus. And this one is called as nuclear membrane. This one is called as nuclear membrane. This cell is called as eukaryotic cell, which is present in from protista to animalia. And prokaryotes are the one which doesn't having this nuclear membrane. Then the cell will looks like in this manner, that it doesn't having any nuclear membrane and it is not compartmentalized. Then this type of cell is called as prokaryotic cell. So prokaryotic cells are present in mostly one kingdom, that is in Monera. They are present in Monera. The and the Monera, the most important prokaryotic example is bacteria. So bacteria are the most important prokaryotic organisms which are existing, and for their economical importance in our life and in our industries and during our lifestyle, it makes that there are large number of benefits and harmful things of prokaryotic organisms. So in detailed way we can clarify these things. So prokaryotic structures are there. For a prokaryotic cell there are large number of things are present, like they are having the cell wall, they are having the capsule, they are having the cytoplasmic membrane, they are having the ribosomes, and they are having the pili, flagella. All these structures are present in the prokaryotic cell. But in prokaryotic cell, there are no cell organelles will present. The cell organelles present will be only the ribosomes only. So here it is showing that appendages, what are the appendages that prokaryotic cells have having means flagella, pili and fimbriae. And we will discuss later about each one. And the prokaryotic cell outer structure, prokaryotic cell outer structure, that is, if it is the cell, the outer structure is present for the some prokaryotic bacteria. It we can call it as the cell envelope or capsule, and the membrane behind this capsule is called as cell wall, and membrane behind the cell wall is called as cell membrane. So this is the capsule, this is the cell wall, and this is the cell membrane. And the its cytoplasm, this is the cytoplasm, and this cytoplasm contains ribosomes, chromosome, and plasmids. All these are present in the cytoplasm. Next, basic bacterial morphology and its ultrastructure. Only two types of cells are produced by all living organisms on Earth. So there are only two types of cells in Earth. One is prokaryote, another one eukaryote. Prokaryotes are primitive nucleus do not having membrane bound nucleus. That is, in a prokaryote, there is no nuclear membrane. That means absence of nucleus. And eubacteria are the oldest type of bacteria, and archaebacteria are the ancient bacteria. And second type of organisms that are present are eukaryotes, which are having the true nucleus, having a membrane bound nucleus. That is, it is having a cell and it is having a nuclear membrane, that is membrane bound nucleus. So for this we can consider as algae, fungi, protozoa, plants, animals. For this we are considering only the bacteria only. Okay. Next, by coming to the chemical composition of bacteria. What exactly the bacteria contains? Bacteria contains 70% on water like humans, and its dry weight is 30% composed of DNA, which is of 5% only. Being the bacteria is very small, it contains just only 5% of DNA, and its RNA is 12%, and its RNA is 12%, and consists of protein of 70%, which is included in ribosomes, protein particles, enzymes, surface structures. All this consider under the 70% of protein. And polysaccharides, nothing but the carbohydrates which are present in the membrane like N-acetyl muramic acid, N-acetyl glucosamine, and other carbohydrates which are of only 5%, and lipids of 6%, and phospholipids, most important structures of the cell membranes. So phospholipids are play a major role in the cell membrane, which is of 4%. Next, bacterial surface structure. Bacterial surface structure, the outermost layer of the bacteria. So if it is the cell wall, the outermost layer of the cell wall is called as cell envelope. Nothing but the glycocalyx. Some extracellular material secreted by many bacterial cells in the form of capsule. So cell wall produce some extracellular materials to make this capsule, and it is a slimy layer. This capsule is a slimy layer loosely associated with the bacterium and can be easily dissolved or easily washed off. And this cell envelope consists of layer of polysaccharides, proteins. And what is the main function of the capsule? It is meant for protection, for the being it is the outer region, and identification, what is the main structure of the cell, and vaccine preparation, and it is meant for attachment of the tissues where they are growing, and it acts as a antibiotic barrier, and it makes hindrance and it stops the production of antibiotics that we are producing in industries against to this cell wall. Being if the antibiotic should be act on its bacteria, it should cross it the capsule. So capsule makes it very strong for not acting any antibiotic on it. Next, by coming to the cell wall structure. So cell wall. So we have discussed about this is the cell wall, and already we have discussed about the capsule, the outermost layer of cell wall, and now we are going to discuss about the cell wall. So cell wall is a membrane which consists of peptidoglycan layer. Here peptido means peptide and glycan means carbohydrate, and supports what the peptidoglycan will do. Peptidoglycan support and shape of the bacteria. The cell of the bacteria, that it may be a cocci, or it may be the bacilli, or it may be the vibrio. Everything is depend upon the peptidoglycan structure only, and its cell wall shows its shape. So based on its shape, we can say the bacteria is coccus, nothing but the spherical, which is round shaped, and bacillus, which is rod shaped, and spirillum means which is S shaped. And mycoplasmas are the bacteria that have no cell wall, therefore have no definite shape. The only prokaryotic bacteria which doesn't having any cell wall means mycoplasma, which are very smaller than the bacteria, and these are also be called as pleuropneumonia like organisms. Okay. Next, by coming to the cell wall composition. So already we told that peptidoglycan, which is made up of polysaccharides plus protein. And what this peptidoglycan is made up of, and how these polysaccharides and proteins are arranged in it, are that is repeating glycan chains of N-acetyl glucosamine and N-acetyl muramic acid. A set of identical tetrapeptide side chain attached to N-acetyl muramic acid and a set of identical peptide cross bridges makes this cell wall a composite network like structure. That is, here the structure is like this. For example, it is a cell membrane, N-acetyl glucosamine and N-acetyl muramic acid, again N-acetyl glucosamine and again N-acetyl glucosamine, N-acetyl muramic acid, N-acetyl glucosamine. That is, each one is linked to the another by 4 tetrapeptide, that is 4 amino acids to the opposite one. Here also the it linked to like this, it is linked to like this, linked to like this, linked to like this, and there is a linkage between these two by a 5 amino acids known as pentaglycine bridge. So I will explain this in the next picture, by that you can clearly understand about it. So this is the network structure of peptidoglycan. Here see that N-acetyl glucose amine and N-acetyl muramic acid. That is, for example, this is the N-acetyl glucosamine. This is N-acetyl glucosamine, and this color one is N-acetyl muramic acid. And these all N-acetyl glucose are linear, and all N-acetyl muramic acids are linear. And each N-acetyl glucosamine is linked to the 4 amino acids here. 1, 2, 3, 4 amino acids are here. And from this N-acetyl glucosamine to and this N-acetyl muramic acid, it is linked by the pentaglycine bridge. For example, if N-acetyl glucose having 4 tetrapeptide side chains and here N-acetyl glucose having 4 chains, these two are linked with the pentaglycine bridge structure. So in this way, by forming the dense network, cell wall is a very composite network of the peptidoglycan, which is highly resistant to most of the infection, most of the antibiotics. And this is the area where we are targeting to get the antibiotics for our health purpose. So in the next video, we'll clearly explain about the how the N-acetyl glucosamine, N-acetyl muramic acid synthesis is happen. By that, you can clearly understand about the peptidoglycan formation. Being it is ultrastructure, you may know the basic knowledge of what is the basic composition present in the peptidoglycan. Next, how can we differentiate? There are mainly two types of bacteria. In whole world or whole nature, there are two types of bacteria. One is gram positive bacteria, another one gram negative bacteria. So all the bacteria are divided into two types, gram positive and gram negative. On which basis we are differentiating this gram positive and gram negative means based on its peptidoglycan only. If a gram positive bacteria is having the only cell wall, then during the gram staining reaction process, it looks like purple color. For the gram negative bacteria to the cell wall, it is having the additional layer of another known as capsule. It looks like pink color during the gram staining reaction. This gram staining reaction differentiates the whole bacteria into gram positive and gram negative by using the gram staining process, which is we have done and seen under the microscope. Gram positive looks like the purple color and gram negative looks like the pink color. So basis of gram stain reaction, here gram indicates that the discovered scientist known as Hans Christian Grams in 1884, and this gram staining reaction is also be called as differential stain. So it is going to differentiate the bacteria. That's why it is called a differential stain. So how what it is going to differentiate? It is dividing whole bacteria into gram positive and gram negative cells. Gram positive cells have thick peptidoglycan layer with embedded teichoic acid. That is, gram positive bacteria having thick peptidoglycan layer. That is, it is having the dense network of peptidoglycan. Gram negative cells having thin peptidoglycan, that is, it is having less amount of peptidoglycan layer. Outer membrane is present, that is called as capsule, and it is also be called as LPS layer, that means lipopolysaccharide layer. I hope clear. And this is the procedure of gram staining technique. I will explain you in a clear picture. That is, first of all, take a slide. First of all, take a slide and add some bacterial culture onto it. That it may be, you don't know about it is a gram positive. This is called as smear. This is called as smear. And yeah, to this, first of all, the primary stain known as crystal violet is added onto this smear. Wait for 1 minute and it is subjected to washing. That is subject to it. That directly, you should not wash directly smear. You should add the water at the edge of the slide. Then slowly water will come and the excess stain is going to be removed. What happened during that? Both cell walls will stain with the dye. Later, you will add grams iodine. Grams iodine, you can call it as a mordant. During that, dye crystals trapped in the cells, dye crystals, that is whatever the crystal violet you have added primarily is trapped inside the cells in gram positive bacteria, and there is net effect of 0 of iodine on the gram negative bacteria. It cannot form dye crystals. And in third phase, alcohol, a decolorizing agent. So when you added the alcohol, what happened? Crystals remain in the cell. That is, in gram positive bacteria, crystals remains in the cell. But the gram-negative bacteria is having the outer member of LPS layer. This lipid layer get dissolved. This lipid layer gets dissolved, and it removes the primary stain and gets outside of the LPS layer. That's why outer membrane is weakened. Cell loses dye, and crystal violets remain the purple, and it doesn't having any dye. And finally, counter stain with saffron in whatever the stain you are going to add, it will looks like in the pink color. So red dye retains with the gram negative bacteria. So being it is having, it is fixing with the crystal violet color, it doesn't needs the saffron in, so it will add usual gram positive bacteria looks like the purple color, and gram negative bacteria looks like pink color. So in this way we are going to differentiate the gram positive and gram negative bacteria. So gram positive bacteria, that is peptidoglycan, one second. So cell membrane. So now we have completed about the cell wall. Now we will enter into the cell membrane located just under cell wall, which is very thin. That is, the cell membrane is most important for formation of its transport of molecules to and fro from the medium to the cell, which is also a lipid bilayer similar to the plasma membrane of other cells, which is useful transport of ions, nutrients, waste across the membrane. Where the cell membrane is present, it is behind the cell wall. If it is the cell wall, and this is the cell membrane, and this cell membrane is useful for transport of ions, nutrients, and water across the membrane. And this cell membrane is made up of 30 to 40% of phospholipids, 60 to 70% of proteins, and some are exceptional cases like mycoplasmas, which is having the more amount of sterols, being they are not having any composition of membrane. There are different functions of cell membrane. So there are different functions of cell membrane. What are the functions of cell membrane means? So carries out functions normally carried out by eukaryotic organelles. So all the eukaryotes doesn't have any cell wall. The outermost region is only plasma membrane or cell membrane. Likewise, what are the functions that are having in the eukaryotes? Prokaryotes also be having the cell membrane, which are the functions which are very similar manner. So it is the site for energy function. That is why we are calling it as site for energy functions, means large number of electron transport chain reactions and oxidative phosphorylation is occurring in this membrane only. And it is meant for nutrient processing. That is, cell membrane is useful for the making the molecules outside and getting the molecules into inside. And it is the area where the membrane gets synthesized on its own, and transport of nutrients and waste materials from to and fro through the membrane. And it is selectively permeable. Selectively permeable means the molecules that are useful to the cell are transported to inside unless they cannot be transported. So that's why selectively, that is, it selectively allows the molecules to inside. Most enzymes of respiration, that is glycolysis enzymes, are TCA, or it may be the electron transport chain enzymes, are mostly related to the cell membranes. Enzyme synthesis of structural molecules, cell envelopes, and appendages are linked to the cell membranes. And secretion of bacteriocins, nothing but the collicins, which are which produce toxins and enzymes into the environment from the cell membrane only. So it is encased by cell membrane, dense and gelatinous, and prominent site for biochemical and synthetic activities. And this total cell is of 70 to 80% of water, mixture of nutrients, sugar, amino acids, and salts, and building blocks for cell synthesis and energy. Now we will enter into the bacterial chromosomal structure. So if it is the prokaryotic cell, so it doesn't having nucleus, means it is having the DNA, nothing but its bacterial chromosome, which is called as naked DNA, which is called as naked DNA. That is because it is not surrounded by anything. That's why we are calling it as a naked DNA. And this DNA is single circular strand of DNA. That is single circular strand of DNA. Only single strand which is very circular, aggregated in dense area, and this is the area where this DNA is present in this total cytoplasm. This area is called as nucleoid. Long molecule of DNA tightly coiled around protein molecules. In as usual, like in eukaryotes, there are histone proteins which are linked to the DNA. Here also the circular DNA is linked to large number of proteins. And further, there is a another, here you are seeing the main naked DNA here, and another DNA is present to the this chromosomal DNA, that is extra chromosomal DNA. So here it is there. Why it is called as extra chromosomal DNA means this is chromosomal DNA and this is extra to this chromosomal DNA. That's why you are calling it as a extra chromosomal DNA and nothing but plasmid. Nothing but plasmid, which is very tiny and circular. Here I am showing that it is very tiny and circular. Free or integrated, it may be present as free, or it may sometimes integrated with the host chromosome and duplicate and are passed on the offspring. During the prokaryotic cell division, during the binary fission, this plasmid also be enter into the another cell, and it can also make the its own copies in this cell only. And this is the most important one, plasmid, that is useful in genetic engineering studies. Types of plasmid. There are different types of plasmids that we are going to discuss. For example, fertility plasmid. They are capable of conjugation, transfer of genetic material between bacteria which are touching. For example, if there is two bacteria present, there is a transfer of DNA from this to this by using the a tube known as conjugation tube. So this during this the fertility type of plasmid transfer its DNA to the other bacteria, and this process is called as conjugation, which are directly linked to one another. This type of plasmids are called as F plasmids. And second one, resistance or R plasmids, which contains genes that can build a resistance against antibiotics and help bacteria to produce villi. And another type of plasmids are known as resistance plasmid. That is, they are resistant to the antibiotics. That is, one bacteria is producing a colicin, nothing but the antibiotic to kill this bacteria. To sustain to this antibiotic, bacteria is producing resistant molecule protein molecules which can stop this killing of this bacteria. This type of protection is given by the R plasmids. And third one type, coal plasmids, which contains the genes that determine the production of bacteriocins, proteins that kill other bacteria. As already I told that, and it is the plasmid, it can also produce the things known as colicins, nothing but who are later called it as antibiotics, which are used to kill another bacteria. Next, degradative plasmids. which enable the digestion of unusual substances. That is, if there are unusual substances are present, which can be degraded using these degradative plasmids. And another one is virulence plasmid, which turn the bacterium into. So, if the host chromosomal DNA doesn't produce any pathogenic features, but sometimes these plasmids produce some substances which make it virulent and pathogenic organism. So, in this type, there are different types of plasmids are produced in the bacteria. Next one, cell division in prokaryotes. Likewise, in eukaryotes, there are different types of reproductions, like vegetative reproduction in plants and asexual reproduction and sexual reproduction. But bacteria is having only one type of reproduction, that is called as binary fission. That is one cell divided into two. One cell divided into two. Prokaryotes use a relative simple form of cell division known as binary fission. This diagram of one shows a bacterial cell. This is the bacterial cell. The cell wall and the membrane are in the red. The cell wall and membrane are in a red. The bacterial chromosome is blue. The cytoplasm is in light green. This is the cytoplasm. This is the cytoplasm. The yellow dot represents a point of attachment of chromosome to the cell membrane. That is, this is the area where chromosome is attached to the bacterial membrane. During this process, one cell become two, having two DNA molecules, and two DNA molecules get separated and becomes two different cells. In this way, this process of division is called as binary fission. In most of the bacteria, it is completed in 20 minutes. So that's why bacteria prokaryotes are used as the most important organisms in the industrial purpose for production of large number of drugs and antibiotics and some useful proteins. And already we have told that the prokaryotes doesn't having any cell organelles. They are having the only cell organelle ribosomes, which are meant for protein synthesis. That is, if is a cell, if it is having its chromosomal DNA, from DNA we will get what? RNA. So RNA are the platforms which is captured by the ribosomes and which are meant for synthesis of proteins in this to sustain the for the division of the bacteria into 1 into 2 also requires the ribosomes, which are the factories of the protein synthesis. And in ribosomes there we are having two types of ribosomes, that is 70s and 80s. 80s ribosomes are present in the eukaryotes, but bacteria is having 70s ribosomes. And the 70s ribosomes are divided into two types. One second. And these 70s ribosomes are divided into 50s and 30s. But in eukaryotes, it is divided into 60s and 40s ribosomes. Here 50s is the bigger subunit, 30s is the smaller subunit. Here 60s is the bigger subunit, 40s is the smaller subunit. And to that additionally it having some inclusions to store the food material. So here one way different bacteria store its food materials in the form of different inclusions. If nutrients abundant stored intracellularly, they are called as granules, and crystals of inorganic compounds not enclosed by the membranes. What are the stored materials? Polyphosphate in Corynebacterium, sulfur granules in photosynthetic bacteria, and metachromatic in the mycobacterium. And by coming to the another one, bacterial internal structures. Bacterial internal structures are known as endospores, which are inert resting cells produced by the gram-positive bacteria, that is Clostridium, Bacillus, and Sporosina. And as already we know that spore is very resistant, and the spore formed inside the cell. That's why we are calling it as the endospores. And already we know that spores are very resistant and they are formed during the unfavorable conditions. If there is no favorable conditions to grow, they form endospores and become resistant to years to years. They are having two phases of vegetative life cycle. Vegetative cell, metabolically active and growing. So during the endospore formation, they first of all they form vegetative cell, which is metabolically active and growing. And second stage is endospore. When exposed to adverse environmental conditions, capable of high resistance and very long term survival. Features of spores: size, shape, and location, and identification. So, and the process of formation of these endospores is called as sporulation and formation of endospores. Hardiest of all life forms, spores are very resistant. Forms inside a cell, functions in survival. Not a means of reproduction. During the spore formation, it doesn't undergo any reproduction. Withstand extreme temperatures of heat, drying, freezing, radiation, and chemicals. So, no, these effects will harm this organ, that is prokaryotic cell, during the its endospore formation. When the conditions gets favorable, it becomes again into the individual prokaryotic cell. And external bacterial appendages. External bacterial appendages. These external secretal bacteria include first one, pilus. That is, if it is a bacterium, it is having small amount of appendages called as pilus, only found in gram-negative bacteria, tubular, hair-like structures of protein, larger than more than in Fimbriae. So Fimbriae are the another type of appendages. For that Fimbriae, when compared to Fimbriae, pillae are somewhat more larger. And here two types of pillae are present. They are attachment pillars and sex pillars. Attachment pillars are meant for attachment to the where they are growing in the medium. For example, we are having our legs to reach with the to touch the ground. In the same way, the pillae are useful for the attachment of the organism. And sex pillars is meant for the transfer of its DNA from one bacteria to the another bacteria. So pillae are useful in the helpful of transfer of one bacteria to the another bacteria its DNA by the process of conjugation. So these are the other type of appendages we will learn individually. Fimbriae, which are smaller than the pillae, adhesion of cells to and surface. It is meant for attachment to the surface, that is to bacteria to attach to its surface. And it is also important for formation of pathogenesis. And the most important appendages are flagella. These are the most important, that is, it is responsible for movement of the bacteria. Long appendages which rotate by means of motor located just under the cytoplasmic membrane. Bacteria may have one or few or many flagella in different positions of the cell. So, if it is the bacteria cell wall and this is the cell membrane, this flagella will arise from this cell membrane. And these are the important organ appendages for the helpful in the movement of the bacteria. It is this flagella is a most important network process having a small motor in its cell membrane, and this flagella is made up of a protein known as flagellin. And based on the type of flagella and a amount of flagella and position of flagella, there are different types of bacteria. Some flagella having one type of flagella, and some bacteria having two flagella, and third some types of bacteria having whole body is filled with the flagella. Based on the position and based on the number, we are differentiating these bacteria into different types. Advantages. So, being the flagella is present, if there is a chemical, it is moving towards this chemical by using this flagella. So, this type of process is called as chemotaxis, moving towards the chemical, and if it is a chemical is harmful, it is going away from the chemical. This type of process is called as positive chemotaxis and negative chemotaxis. And the flagella main function is for the motility. Next, types of bacterial motility. So, we can run and we can walk. In this way, flagella is useful for the types of bacteria, that is, it can run or swim. When a bacteria moves in one direction for a length of time, it is called as running or swimming. And it tumbles, periodic, erupt random changes in direction. If you want to change in direction, this process is called as tumbling. And swarming, rapid wave-like growth across on a solid culture. If it is a growth of medium, there is a rapid growth of medium. That swarm-like structure is called as swarming. Mechanism of flagellar movement relative to the rotation of rings in the basal body of the plant. So already I told that the cell wall, that is the cell wall inside is a plasma membrane. There contains a motor type of network which contains a large number of rings. These rings are responsible for this flagellar movement. And antigenicity, flagella or H antigen is useful in the serological types of organism. To identify the which type of bacteria is it is, by the antigenicity of it, we can know the which its pathogenicity. And already I told that based on the position of the bacteria, based on the position of the bacteria and number of, sorry, no, I'm not, based on the position of flagella and in time number of flagella, we are dividing the flagella bacteria into different types like monotrichous. Here means bacteria having one flagella at one end. Lophotrichous, bacteria having flagella at one end which are very tuft. Amphitriching, it is having a single flagella at both ends. Peritrichous, having flagella at whole body, is called as peritrichous. So, how can we see that the bacteria is motile or not? It is done using a microscopic experiment known as hanging drop experiment, where a slide is present on which we are going to take a smear which is having small amount of cavity, in which you are adding the smear to this. Yes, you are putting a cover slip, and there you are adding some gel or some drop of water, or it may be anything to for the movement of this bacteria, and we have taken this slide into the microscope. In during the microscope, if the bacteria is moving like this movement is due to the flagella. It is clearly seen in this drop of liquid you have added for movement. This type of one is called as hanging drop preparation for the evidence of motility. So these are the basic differences that we have discussed in the previous slides, and this is a slide showing the all the differences of prokaryotic and eukaryotic organisms. So I will explain very briefly. So size of the cell typically 0.2 micrometers in diameter, which is very small, but eukaryotic one having 10 to 100 micrometers. No nuclear membrane, but it is having the true nucleus. So we already that prokaryote is having no nucleus, but eukaryotes having clear nucleus. Membrane enclosed organelles absent. There are no membrane uh, cell organelles. It is having mitochondria, Golgi complex, endoplasmic reticulum, ribosomes, everything. It is having only ribosomes. Flagella, it is having the flagella like structures, contains two protein building blocks, and it is having microtubules in its membrane. It is having the glycocalyx, that is a capsule like layer outside the cell wall. It doesn't having any glycocalyx. It is having the cell wall, that is it is having the cell wall like structure that is present on the cell membrane. It doesn't having any cell wall. The final outermost membrane is the cell membrane. Plasma membrane, no carbohydrate, generally lacks sterols. It is doesn't having the carbohydrates and lacks sterols. It is having all the carbohydrates and sterols. Cytoplasm, no cytoskeleton. That is, in prokaryote, there is no cytoskeleton. That is, no microtubules are present to make it stiff. But the eukaryotic cell is having the microtubules towards in the cytoplasm. Ribosomes are 70S and ribosomes are 80S. Chromosomal DNA, which is a circular chromosomal DNA. Here, multiple chromosomes in nucleus, there are large number of chromosomes are present. Cell division by binary fission, that is, one bacteria is divided into two very fastly for 20 minutes. But here the cell division is by mitosis process. Cell division is by mitosis process. And sexual reproduction, there is no meiosis, just transfer of DNA to its respective one. But here sexual reproduction is formation of the gametes, and it is done by the meiosis process. These are the basic and final differences of prokaryotic and eukaryotic organisms. Thank you.